This is Ahmed Ghanemi, the team leader of AUC Egypt iGEM team, and together with Rana Salah. Introduce yourself.
I'm Rana Salah. I'm a biology major at American University in Cairo, also like Ghanemi, and we are doing a minor in bioinformatics, and we are actually leading the team this year for AUC Egypt, and we have also our great instructor, Dr. Hannah.
Hi, I've already introduced myself, but yeah, I'm Hannah Abdulzahir. I'm the team's instructor, and we're working together with our PI, Dr. Anwar Abdel Nasser from AUC Egypt. He's the director of the Global Health Institute at AUC.
Thank you for introducing yourselves. I'm Josemary on behalf of Molecular Cloud. And today we're interviewing AUC Egypt iGEM. So please, if you could tell us more about your project involving developing COVID-19 therapeutics.
Yes, so let me share my screen.
Sure, 1 second.
So as you can see from the title, it's a SynBio-based SARS-CoV-2 therapeutic solution for SARS-CoV-2, basically. Orchestrating inducible interference using virus-like particles, toehold ribose switches, and short interfering RNAs. So let me start with a brief animation to get into the full picture of our project. So descending from the left is a virus-like particle, more specifically a SARS-CoV-2-like particle that's expressing the spike protein, and hence it's capable of targeting cells expressing the ACE2 receptor, the receptor for SARS-CoV-2. So once inside the cell, and once our circuit gets into the nucleus, a toehold ribose switch is constitutively expressed. This toehold ribose switch is our sensing moiety. If it finds SARS-CoV-2 targets, it unfolds, exposing the ribosomal binding site and the start codon, allowing for the expression of a downstream activator. This activator in turn induces the expression of short interfering RNAs that are capable of targeting the replicase region of the virus, thereby inhibiting further growth and interfering with the replication cycle of SARS-CoV-2. So before I get into the circuit, let me introduce you to the toehold ribose switches. And this figure is retrieved from the original publication by Green and colleagues. To the left here, you see the ribose switch, the toehold switch, which is composed of, to the right, is the repressed gene. And why is it repressed? Simply because the start codon, the AUG, and the ribosomal binding site are hindered by this stem loop. But to the left of them, we have the toehold domain, designated here as A and B. A and B are complementary to trigger RNAs. If the toehold switch finds the trigger RNAs, they bind to it and they expose the ribosomal binding site and the start codon, and hence activate the translation of the downstream gene. So for the design of the switch, it's a little bit challenging because you design, but you can't predict the actual behavior in vitro or in vivo. But simply, you start by searching for triggers in our case, we're searching for triggers in SARS-CoV-2 mRNA. For these triggers have to be accessible, i.e. there are lower secondary structures around these regions for the toehold to be able to bind to these triggers. And second, they have to be orthogonal. They have to be unique to SARS-CoV-2. In our case, we don't want them to be absolutely unique. They just have to demonstrate orthogonality to the human genome. And third, they have to demonstrate lower mutation rates in order to maintain the efficacy of the drug and prevent failure due to mutations later on. So once you identify the trigger, you start building your toehold switch. The original toehold switch was designed to be expressed in prokaryotic systems or cell-free expression systems. But here we're making a little bit of tweaks to make it suitable for expression in mammalian systems. One of the differences is that the loop up here, you don't find this bulge, the bulge of the start codon, because the ribosome binding site in mammalian cells is directly attached to the start codon, and hence you'll find them all in this loop. So you first construct the toehold region, which is complementary to the trigger. They are about 30 nucleotides long, and then you add the ribosome binding site in the start codon in this loop. And then once you make the switch, you check for the presence of stop codons to ensure that there are no stop codons after the start codon, and hence efficient expression of your target gene. You then perform some energy calculations to check the stability of the toehold rib switch by we utilize an OPAC software, which you feed it a sequence of RNA and it gives you the free energy calculations of it. Then you perform free energy calculations with the sequence and the triggers to predict if they can bind efficiently in vitro. But the predictions most of the time fail because predictions based on free energy are not that efficient. You design as many toehold switches as possible, and then you test all of them in vitro and find the best performing toehold switches. So let's get into the circuit. The circuit is deliberately designed to express the toehold switch, the sensing moiety, constitutively, and hence we chose the CMV promoter, which is a constitutive mammalian promoter. It's a strong promoter. Once inside the cell, the toehold switch is expressed constitutively in large quantities, and if it finds trigger, it unfolds giving rise to Gal4VP16, which is an activator. This activator is a potent transcriptional activator, and it's orthogonal to mammalian cells. So this is to ensure that there are minimal to no crosstalks in our system. The Gal4VP16 basically has a downstream promoter, which is the UAS. This UAS controls the expression of our short interfering RNAs, which are targeting the replicase region of SARS-CoV-2, thereby inhibiting the growth and interfering with the replication cycle of this virus. So that's it for the circuit. We performed mathematical modeling using SymBiology software of MATLAB. This is a basic schematic of it. We did most of the equations are this is the layout of the modeling, but we're in this stage we're adjusting the transcription and the expression constants basically to finally predict the levels of short interfering RNAs and if they are going to be capable of inhibiting as much mRNA of the virus as possible. So now Rana can continue with the delivery method, which is stage 2 of our project. Let's stop sharing. Rana, you're muted.
Okay.
Okay, just uh, 1 minute. Okay, now I'm gonna tell you more about the virus-like particle systems. We are actually using it as a delivery system for our project, and we mainly used it for the following advantages. We have first, we wanted to be specific targeting for the cells expressing the ACE2 receptor in order to like move our, to inject our toehold switches and sRNAs only to the cells that can be affected by the COVID-19 or can be affected by SARS-CoV-2, not to all of the cells. And it's also an important part of the virus-like particle system that they lack the viral genome and they are not replicating particles, so they will not cause another infection for the affected cells or whatever other unexpected combinations. Also, it can escape the lysosomal degradation. And furthermore, I want to tell you more about how we are gonna produce our cells. It will actually be the second phase of our project. And after the first part, we will be testing how the toehold switching and sRNAs can be working together. And for the second phase, we will be producing the virus-like particles alone without loading the circuit. In this phase, we will actually be using a lentivirus pseudovirus based on lentivirus. We will actually use 4 plasmids. The 4 plasmids, they will be the first one. We are modifying on a work of developed by Catford 2020, and it actually the modification we are doing for the lentivirus systems that we are not using the TAD board, because in our system we will be only using the other plasmids, one for a GEG and ball, and the other for the direct board, because the TAD board we will no more be using it. As I will view for you in the next slide, we will be using a chimeric 5' LTR, and it will help us to start the with the help of a heterologous promoter. It will help us in order to start the transcription of the injured gene without the help of the transactivation by the TAD part of the virus. The second part, we will be having a plasmid for spike, and I will talk more in the second slide also about the different versions that we will have for this spike. We will have 3 different versions. And finally, we will have for the third phase, we will start the loading of 2-volt switches and sRNAs into our plasmid. So I will tell you more about the structure of the backbones that we will have. We target that these pseudoviruses will be able to target only the cells showing ACE2, and we will have a part for luciferase iris that is green. It's like any or any reporter gene in order to mark if the infected cells were actually that it truly was infected. And then for the second, for part of the second phase, also for the different versions that we will have for this bike, we will have the first part is the normal codon optimized spike, and it's actually codon optimized for the baculovirus system. They'll tell you more about why we're going to use it. The second part is called spike ALA-YT, and it has mutations in its cytoplasmic tail. The reason that we have these mutations in order to make it better for the plasmic membrane expression, because it will lack the endoplasmic reticulum retention board. And also we will have a third one, which is spike HA tail, and it replaces the cytoplasmic tail of the coronavirus with the one for influenza, in order to give it to test if it will be better in pseudotyping, because in the literature it was more efficient in pseudotyping. And then for the third phase, when we are going to add our gene of interest into the whole plasmid, As I mentioned, we will have the 5' LTR in order to replace the tag port, and we will have the 3' LTR, and then we'll add the gene of interest between them, and the other packaging plasmids that we will have from the lentivirus are the ones, the two plasmids I covered before, and also it will only show, we won't have the envelope part for the lentivirus because it will show the spike protein for SARS-CoV-2 in order to be a decoy particle for it. We will be using the virus expression system using a co-expression, which is the one on the right, as we will be having 4 plasmids, and we will form our VLPs mainly by the disassembly and reassembly, which takes place normally in the virus expression system. And we mainly choose the system in order to magnify our production. And now, actually, you may be thinking about how the safety will be good for our project and can be included within it. So we actually like using the both plasmids for the part of the lentivirus without the tag port. It will help us to use the third generation of the expression of lentiviruses, the pseudo ones, in order not to cause the amplification or replication, because it will have it will normally silence its replication. And then we are also not only working on it from the lab, we are taking the synthetic biology applications out from the lab to the web. And that's what we are working on for our human practices. So first, we are doing a crisis simulation related to especially the biosafety and biosecurity parts. And we are giving these sessions like workshops for students from STEM schools, and they are high school students, and they are usually doing capstones and projects that deal with different challenges that are faced by the web. So we are giving them more information and about biosafety and biosecurity and how to take more decisions related to this part. We are also having a same biology game, and I will show you a small video for the interface of the games that we finished. This part is mainly targeting the science communication. So we are using it to science communicate the different topics in biology and SimBio to people usually from the preparatory stage to undergrads in a more interactive platform. And we are also working on developing GMOs awareness. That depends on the surveys that we will be conducting, and based on the results that we are gonna get, we are start doing the GMOs awareness, understanding more about the society and how they view it. And we actually started that, and we had the session and in hot prize competition. It's more, if you heard about it, it's more of a competition that targets different challenges each year, and a lot of startups are actually launched from this competition. We are also maybe working on a virtual patient simulation, and we had also, I will show you the interface for it. It's actually a simulation that targets both raising more awareness about the symptoms of COVID-19 for the usual public, and mainly for doctors who will be dealing with cases of COVID-19 and how they can deal with it. And for the part of collaborations, we will be having 2 collaborations. Mainly, we started their ideas from what we learned from quarantine. So for the first one, it's an SDGs from home. We want people to understand that they can start advocating for a certain SDG and working for it from home with very simple tools and very simple practices. They don't have to start the mega projects. And for the Thank You Winter, it's be instructors and how they helped us in order to reach and developing our project in these times. I will stop sharing and show you the board for the video. Okay, give me 1 second. Is it shared now?
Not yet. Can't see the video.
It's mainly a case where fireflies are trying to fix their daughter fireflies that belong
Ron, the
to
video is not
repositories.
running. Excuse me for interrupting.
Really? Oh.
Video, yes.
Oh. So,
Might take a while to load.
And share instead of sharing the entire screen, you may share the window for the video itself.
it's not giving me the option to do this actually. Okay, so Magdi, can you try to do it? I sent it to you.
I think it will take a while to
You'll find
download.
them.
So, so I'll share screen now.
Sure.
Video. So the purpose of this game is we wanted to make, yes, we wanted to introduce high school students to the principles of synthetic biology in a story format. This is a firefly whose luciferase gene is mutated for some reason, and they go to the SynBio lab to fix this mutation. And it's full of challenges, and the beta version will be released soon. Okay, now I can stop sharing. So this was a brief of our project. We would like to thank you so much for hosting us.
Thank you so much for that presentation. It was so thorough, and we definitely think your therapeutics idea is extremely innovative. I do have a couple questions before we conclude. So you mentioned that orthogonality is an important part of the project, especially with the toehold switch. Can you explain why orthogonality, especially to the human genome, is so important?
So that's a very good question. You need to have your toehold switch orthogonal. They need to bind only to SARS-CoV-2 targets. You don't want them to get activated because they share some sequences with the human genome. That's where you're going to get some false positives. That's it.
Very good. Yes, I definitely think that false positives right now are not very conducive to our data collection, especially for such an unknown virus. I have another question. So you showed us the circuit that you were developing. So how did you identify these targets? For example, Gal4. How did your team work on identifying these targets?
We basically started searching for promoters that are also orthogonal to the mammalian an orthogonal promoter to mammalian system, and we wanted some potent transcriptional activator, and we found Gal4VP16 in the registry of standard biological ports. That's how we did it.
Okay.
And the same goes for CMV. CMV is considered a promoter that's also orthogonal in mammalian systems, and it's a potent transcriptional, it's a potent promoter, mammalian promoter.
Okay, okay. And you also mentioned that your team is using MATLAB to use the mathematics to create your schematics and carry out your design and methodology, essentially. So can you give us a little bit more insight into, you know, how your team has approached this and developed this, and you know, did the pandemic have any influence on your original design versus what your design looks like now?
Yes, sure. Actually, the MATLAB gave us a very good environment for working. We first started sketching the equations manually, and we wrote a code to display how the species interact with each other. But MATLAB and SimBiology gave us a very interactive way of laying out the species in each and how they react with each other. You simply draw lines, and it also specifies the equation and the model that you want to try to use. We're basically basing our models on mass action kinetics and Michaelis-Menten modeling for gene expression systems, and they're all present in the SimBiology application. We started with one big question, which is is our circuit going to produce enough short interfering RNAs that can interfere with the replication of the virus efficiently? And that's what we're trying to answer right now. We're in the process, as I told you, of adjusting the transcription constants to get better predictions of how the circuit will behave once inside the cell of infected patients. I wish that answers your question.
Yes, yes, it does. Is there anything else that your team would like our audience to know about your efforts, your team, your project?
We're still in the design process of a novel therapeutic circuit for COVID-19. We didn't do any actual testing in the lab. That's one of the effects of the pandemic. We had no access to the lab, but thanks to the iGEM committee this year, they allowed for phase 1 projects, which is heavily based on modeling and design, giving us the chance to participate. And actually, this is our first time to participate in iGEM, so wish us good luck.
Definitely. That's really great that you guys
But we have an experienced instructor. Yeah. Hannah Desire is very experienced in iGEM. She's now an iGEM judge.
Awesome. Very cool. Well, AUC Egypt, iGEM, thank you so much for meeting with me today. We definitely enjoyed your presentation, and we definitely want to wish you the best of luck in the iGEM Jamboree. Congratulations on your first year of participation, and we really hope it goes well.
Thank you so much.
Thank you. Thank you for meeting with us. Bye-bye.